Welcome to The Microscopists, a bite-sized bio podcast, hosted by Peter O'Toole, sponsored by Zeiss Microscopy. Today on The Microscopists.
Today on The Microscopists, I'm joined by no other than Brad Amos, Emeritus Scientist at MRC Laboratory of Molecular Biology and Visiting Associate up at Strathclyde. And we discuss failing to retire.
I'm like most scientists. I don't really retire. It's not a desk job. It's a mind job, and your mind keeps you a scientist.
His really varied career trajectory.
My career has been a kind of vertiginous series of switchbacks.
His legendary lab skits.
There were some very chaotic performances.
And the importance of keeping your hands out of the way when scuba diving.
We sat, Linda and I, on a sandy bank at a depth of about 5 meters, and he was waving these fish about, and these reef sharks came around.
All in this episode of The Microscopists. Hi, welcome to this episode of The Microscopists. I'm Peter O'Toole from the University of York, and today I'm joined by Brad Amos from, Emeritus MRC, the Laboratory of Molecular Biology, and part-time at the University of Strathclyde. So I would say retired, but definitely not retired, are you?
No, I'm like most scientists. I don't really retire. It's not a desk job. It's a mind job, and your mind keeps you a scientist. But I'm unusually lucky because I've got a house which I've equipped over the years with 3 workshops for mechanical and optical work, so I have the equipment to carry on to some extent, except I can't quite manufacture a wet lab, so I can't really do as much staining and so on as I used to.
Yeah, and actually supplying a wet lab, even the optics, it's not a cheap game.
Right.
So I presume funding is more limited, except your collaboration maybe with Gail McConnell at Strathclyde and the funding.
Yeah, that's the key thing, and that means I can stay in the swim. And that has been enormously valuable.
So I should have pointed out, actually, to those who are listening on whether it be iTunes, Spotify, Google, Amazon, RSS, whichever platform it is, that actually Brad has sent through some really entertaining pictures as well as serious pictures. So actually it's worth just skimming at least the iTube channel. But the first one I'll show, because we just talked about Gail. So here you are with Gail, which I presume is in Strathclyde itself. Is that correct?
That's right. And the instrument on the right is the Mesolens, which has been built into a confocal microscope by Gail, which is a fairly ambitious and successful engineering operation. The original Mesolens, as we designed it, had no eyepieces, and was very difficult to change from confocal to camera mode. And so Gail has basically taken all of that on. Her colleague, John Dempster, is a person who's supplied software free to all the electrophysiologists in the world. And he's made a software for the Mesolens as well. And the lens designer, Ez Reed, is one of the top lens designers in the UK. So he's a key person as well. But he works with classical optics with dispersion of the ordinary type. And so Gail can get together with him and tell him about nonlinear optics and what you need for a multiphoton system and so on. So it's not only a comprehensive team, but it's an extremely rare team that I've been lucky enough to be associated with.
Now, I've jumped in almost to today instead of going from the past to today, but actually the Miso lens is very famous in the microscopy world. And anyone who is listening that isn't familiar with the Miso lens, you may have used your school microscopes or microscopes at university, and the lenses are typically thumb-sized, maybe, and broader. They're not significantly large, but the Miso lens is, go on, Brad, you'll know the dimensions of a Miso lens.
Well, the largest elements are about 64 millimeters in diameter, and the total length of the Miso lens is about 50 centimeters. So it's as long as an arm. And I do have a photo, which I should have sent you, which is the complete Miso lens dwarfing an entire 1890 brass microscope. So it's huge, and it has to be huge to achieve the optical effect that we want, which is a very large field, 6 millimeters in diameter, and also very high resolution. Obviously, you can look at a large field just with an ordinary stereo microscope, but you can't have resolution so high that you can't see it in the eyepieces. And that's what we were aiming at, because all the objectives that people have used at school and in universities and other places have been basically the same for 100 years, in that they have been designed to suit the human eye. But there's no reason to stay with that restriction now that we have cameras that have an angular acuity greater than the human eye looking into an eyepiece. So the idea of the Miso lens is you can get in one shot what would ordinarily have to be put together by tiling and stitching the area with normal objectives of similar numerical aperture to the Miso lens. But the problem with it is that none of the microscope manufacturers are interested in manufacturing this, because it doesn't connect to any of their existing equipment. One major company, which shall remain nameless, has told me that they are watching, and if Gail and I are successful in getting sales of the Miso lens, they will simply make their own. They won't pay royalties and the reason given was that their engineers are too proud that someone has exceeded them to admit that someone exceeded them in their own special technology. And so we know where we stand. If we succeed, we fail. But the converse is not true.
But if you succeed, there'll be other companies that would like access. And there's actually the field, the type of biological questions that this can address would be wide and varied. There's lots of other companies, non-specialist optical companies, that would very much like to get into this type of field as well, especially with the spatial omics technologies that are coming out now that need that large field of view for area selections. So there is potential. And for anyone who's scared about the size of the lens, I think Brad said sort of the size of the arm, think about a telescope. If you're as a bird watcher, a telescope, it's no different to that, really. So it's kind of just different, not wrong. And I think that's a key take home. But it's been good fun to watch it, and I really look forward to seeing the next developments of it. And I think Gail here has to be a guest with us at some point on the Microscopists, because she's very sharp.
Yes,
and also very
yes, and she has now many projects which have, some are independent but of the Mesolens, but others have arisen, and she's discovered lots of things that we didn't know before. So, for example, her group have found that bacteria in colonies growing on agar actually create nutritive channels and suck in nutrients. So in other words, you can have a prokaryotic cell, and yet it can have organs by cooperation. It can develop a vascular system. And it needed the Mesolens to actually see these rather subtle channels. And so another big project is to combine the Mesolens with the light sheet principle, so that we can have the high resolution, the one-shot acquisition of a huge amount of data, but we can have the speed of a camera. So we have the massive parallelism of a camera, but then the thing is that the camera has to be a very, very special one, because we really need something like 150 megapixels to capture everything in the Mesolens image.
I want one of those on my camera, but they haven't done one yet. It'd probably be bigger than that, actually, as a chip. The chip size would probably be larger than the whole camera.
Yes. Well, the ones that Gail has found and uses at the moment have a fairly standard size chip, but it has a piezo driver which moves the chip around to 9 different positions. And so you get 9 images, and you can work out the equivalent of having 9 times as many pixels in the final result. But that slows the thing down, and that's exactly what we don't want to do.
Yeah. So actually, just there was something interesting you said actually about Gail studying what she's found with bacteria on agar. Gail is, if I'm not wrong, a physicist by training, and a pioneer in. She was one of the first professors, I think, of physics, certainly at Strathclyde, I believe. So trailblazing there. But you yourself are not well, I don't think, but weren't you a zoologist to start with?
Yes, that's correct. I've done lots of different things, and my career has been a kind of vertiginous series of switchbacks in which I venturing into zoology was perhaps rather a mistake, but I made many mistakes, and each time I changed. I morphed into something different, and I was always interested in optics, and I had to learn optics extremely quickly, and I had two advantages. One was that when I started doing research in zoology, the professor of zoology at the time had just been appointed, brought to Cambridge a collection of very, very fancy microscopes from Copenhagen. And we quickly had a rapport, and I was the only person who was allowed to use this Aladdin's cave of microscopes. So I learned about fancy interference microscopes and so on. And then when I managed to team up with John White, which happened during one of the worst dips in my switchback career, I worked with his system and brought a more reasonable microscope anatomy to it. But when we were already in negotiations with a manufacturer, and when the world was beginning to become very curious about what we were doing, and when we had a paper in J-cell biology, which was creating a stir, I started to look at double-stained preparations, and I found it didn't work. The two images taken with the stains of different colors actually had different magnifications, so the two images didn't superimpose. And the problem was that in the optical system that we'd built using improvised lenses and not really knowing much about optics, we had chromatic aberration. And I had to act quickly. And I had a eureka moment, which is very rare in science. I happen to have some concave mirrors, and I don't know what it tells you about my psychology, but I had the habit of idly looking at my own eye with a concave mirror, because the iris is a very intricate and interesting structure. So I was looking, and then I tilted the mirror, and I was pleased to see that the resolution in the visible image was good, even when the angle was so great that I was beginning to see my eyebrow. If I went further and tried to look at my forehead, it was hopeless. The astigmatism and other aberrations began to kick in. So without any knowledge of optics or any modeling in a software, I suddenly realized that I had the basis of a high angle scanner, which is what we needed, because we were making what John White called the shoebox. We were making a box which would fit on to any microscope, would look through the resident eyepiece and would scan, but that meant the scanner had to do plus or minus 20 degrees, 40 degrees total scan at least. And that is quite exacting so far as optical quality is concerned. So I put together this scanner, and you see at the bottom, there is the eyepiece, and then the two vibrating galvo mirrors are between two concave mirrors, which are the discs at the bottom of the diagram. And that worked extremely well. I didn't really model it. I just, I happened to have acquired, mainly out of bloody-mindedness, because I was at one of the dips in my career, a small lathe and milling machine. And I made a rig to hold concave mirrors and two plane mirrors to represent the galvo mirrors. And I made it totally adjustable. And I rushed into the lab with it, and John White and Mick Fordham, who was the head of the workshops in the LMB at the time, just grabbed it out of my hands and began to work with it. And within a couple of hours, they discovered that if they adjusted the two mirrors, they could get linear scan lines from a laser. And this was in retrospect, it didn't seem so surprising, but later on I discovered that this was a known proposal which was in prior art in the literature for a scanning system, but in the words of the patent attorneys, the author had taught away from this, saying that such a scanner was useless because it gave curved scan lines. So, between us, the three of us had solved that problem. But I took out about 5 optical patents, which were granted, and a few more that didn't get granted. And that particular one for the scanner didn't give us any protection, because when the big microscope companies started making confocal microscopes, they didn't have to use their own eyepieces, because they could substitute a lens of long focal length that had the right chromatic correction and so on to suit their objectives. That's secret information. We couldn't do that.
Yeah.
But they did it, and it meant they could have a scanner of a much simpler type, which was just basically 2 galvos close together and a long focal length lens, instead of the eyepiece, which had a focal length of only about 30 millimeters, usually. So I was in optics by necessity, and I had to learn very quickly what to do.
So I love that this is branching off in all sorts of directions, because you mentioned a low point, and one of the questions I always like to ask is, what was the lowest time? What was the most difficult period that you had? But before we go there, obviously you're pioneering in your optics with John White to get to there, spun out into become the Bio-Rad confocal microscopes, of which one of the last generation was the Bio-Rad radiance systems, which actually was my first confocal. So I have very fond memories of my radiance. I loved my radiance.
Right. Well, I towards the end of the period with Bio-Rad, I decided the radiance was a good design, but that if we were clever, we could reduce the build cost enormously, and the sales price could be about 1/3 of a confocal microscope, so even cheaper than the radiance.
Yeah, I remember the cell map.
Sorry?
Was it the cell map?
The cell map. Yes,
yeah, I remember it.
Unfortunately, even though people had started to buy it, they never got it because we were taken over, and the UK confocal business was closed, and so on. So that was one of the lowest points, actually, but it was not the only one. And the one that I'm talking about, that I mentioned earlier, was actually when I had abandoned my early research topic, which was a different form of contractility from muscle, which occurs in ciliated protozoa, such as Vorticella, which is a little pond organism. I had to abandon that because the project became very difficult, and also my patron, in a sense, Weiss Fo, the Danish professor with the microscopes, died, and it was a very traumatic period because his wife was killed in a road accident, and he progressively deteriorated and became a very difficult man, and then he committed suicide. So that led to my having a difficult time. I was looking for a job because I'd lost my patron, which is gross carelessness in a system with a lot of patronage. And I decided that what I'd done wrong was I had worked on something which was not mainline. It wasn't mainline cell biology. Yeah. So I flung myself into a monoclonal antibody project which I could start by sneaking into the MRC lab, which in those days was never locked. You could just walk in, and my wife worked there, so I could get in and out easily. And she gave me some of her bench space to work on. And I had a project where I was helped by John Kilmartin, who was a brilliant biochemist, and also by Bob Johnson back in zoology, who was an expert in cell fusion, and by Yair Argon, who taught me how to make monoclonals, how to fuse the cells, the myeloma cells with the spleen. And I took the spleens out of the mice. I had prepared the antigen. I selected the monoclonals. I prepared screens for the monoclonals by immunofluorescence using synchronized HeLa cells, which involved staying up most of the night, compressing cultures in nitrous oxide according to a very strict time schedule. In fact, I was trying to do what would normally be done by a large research group, all on my own. And I succeeded. I actually got some monoclonals, which had interesting immunofluorescent distributions in the mitotic HeLa cells. They looked as if, well, there were some that were restricted to the condensed chromatin, others were on the microtubules, and not all of them turned out to be antitubulin. Maintaining them, I found that they were slowly dancing away. They stopped producing. The titer of antibody went down and down from the cultures, and the gurus in the LMB, who were the leaders in the world at that time, said, "Oh, bad luck. It sometimes happens. These hybrids, they just lose chromosomes, so that's tough." But for me, that was a very low time. I with John Kilmartin's help, I got a 2-year grant from the MRC, and that was over. So I had no salary, and I had no antibodies and no result from 2 years of very, very hard work. So that was why discovering that John White, who is a tremendous polymath and works on the nematode brain, and is also able to command the very latest in digital and also good old analog electronics. He had realized the importance of beam scanning, set up a confocal microscope using the time base of an old oscilloscope to drive the scan, and he was getting results, and so he agreed with alacrity to my proposition to modernize the microscope and equip it with things like transmitted light, which it didn't have, and we were both very excited. So he used to come towards the end of the day and say, should we carry on with this tonight? And I used to go in and we'd work until midnight, night after night, doing all kinds of crazy things. I needed to find a slide that worked in fluorescence. So I got some microfilm, which was still being produced in those days. And I made a contact copy of a finder slide on microfilm, because the blue stuff in the microfilm is actually fluorescent. So I could use those to get before and after images. So that was essential for the J-cell biology paper that made us famous. It was essential to have the very same cell or the same group of cells with the best you could achieve with conventional optics and the confocal results side by side. And we did all of that, and I used my HeLa synchronized cells as specimens, and I also got my biological cronies to bring many different things, because otherwise we would have only had HeLa and nematodes. So but at that time I thought that I would just be finding specimens and finding microscopes and attaching them to the confocal in different ways. And I was really delighted to make this contribution of the scanning system, because that did actually save our bacon. We would not have been successful with something that couldn't handle double staining.
Yeah, which we've all benefited from, and now not just double staining, but 3, 4, 5, 6.
Yes,
staining.
Yes, exactly.
The LMB sounds like everyone works hard. It is an amazing environment. You've got that sort of hotbed of innovation.
Yes,
working to midnight, but it's not all. And this is where the pictures get really entertaining, because I believe this is you as Robin Hood.
Yes. Yes. Well, as soon as I started on the confocal thing, I changed my penurious state. I got a salary from Bio-Rad, incidentally. So they paid me as if I was an MRC employee. So I was like Voltaire, who lived on the boundary between two countries and could always skip across into one when the other proved to disapprove. And this meant that I could have these quite satirical skits. And that particular one concerned the history of the lab and the succession, one director by another. And on the screen, it has a slightly altered Beatles theme. And the theme was always about the problems, the real problems of being a researcher. And I was always amazed that there was so much talent suppressed in the lab. So I used to think of a theme. Sometimes it was a pantomime. Once it was the Temptation of Faust, based on Marlowe's version. And there were lots of other things. We had one thing based on the Full Monty. And the number of volunteers was amazing, and their abilities were just you would never believe it. There was a Dutch girl who came forward and said that she had been trained in stage sword fighting. So we had a version of Robin Hood, which I managed to twist around so that we had some stage sword fighting, and we had two women fighting with. They were supposed to be kind of ninja warriors, and I made swords for them. I was all too close to these swishing blades, and I was rather relieved when one of them dropped down as if killed, and the Dutch girl then picked up her head and held it up, and it was a head that I had made to resemble the actress. Out of papier-mâché, and the whole audience went kind of, it was a coup de théâtre, and very, the whole thing was just terrific. And I enjoyed it immensely because it always involved going into the lab on a cold winter night and suddenly, out of the darkness, you went into a lighted area with all these young people excited and ready to go on stage. And of course, the people I lampooned or attacked didn't dare object, because they knew they had to keep smiling, even if they resented it. It became so popular that I became fireproof, and financially I was as well. So
I love it.
It was great. And
more places should do it.
People, very senior people, I won't give names except for Cesar Milstein. Otherwise, you would suspect me of doing a typical bit of Cambridge name dropping. But they were willing to act. Cesar was willing to play one of the cleaners in the lab. And I had him come on stage with a bottle of the cleaning spray and say, oh, they're telling me to clean the floor. I don't know how I clean the floor. I just spray it with varnish, because we had all these dirty lino floors which had never been cleaned, but grew glossier and glossier. But Cesar proved to be totally out of control, and he kept coming on stage and spraying the feet of the people on stage. There were some very chaotic performances.
Do you write these skits yourself each year? There's a couple more pictures. So this, I presume, was you as the Pope?
That was from Faust, and I was the Pope, which had it had the Spanish element in the lab, a bit embarrassed because they were good Catholics, and this was long ago. It was not so far from the Franco era, when you didn't say anything about church or government. And they actually changed after that performance, and they became more and more excited to play satirical parts in the skits. At first it was the Spanish women who wanted to take part, but then the men would come and lean on them and say if you do this, no man will look at you, and they bring shame on your family and so on. But I remember one Spanish girl, I made a song for her, which was about how people who joined Cesar's lab invariably became pregnant soon after. And it was in Spanish. And the remedy, la solución, was cinturón de castidad, a chastity belt for all newcomers to the lab. And she, I thought, was going to chicken out at the last minute. So I grabbed her arm and we went on stage together, and we both sang in Spanish about this. And for a year after that, people in the elevator or wherever would start talking to me in Spanish, because they thought that my vocabulary was enough. But it didn't extend beyond cinturón de castidad, which is not terribly useful for marking off a sprightly causerie in Spanish.
And for the next picture, I presume you are still doing these skits, because this looks rather current.
So this slide is from after I had retired. Chris Ubridge, who was a staff member at LMB, took over the writing of the script, and he really restored the comic aspect, because it had been getting a bit gloomy in my time, and he asked me to play Boris Johnson. So I came back out of retirement for a cameo performance. And the issue was that Boris comes on stage to inquire into who are foreign in the lab, and to get rid of them, get rid of the foreign academics. And there's a lot of stuff about, the English student, who was not foreign, was actually played by a Polish guy whose English was impeccable. And he's the one who gets sent off because he says he doesn't actually watch Bake Off. And so Boris then said, no Bake Off, Johnny Foreigner, and has him off stage.
And he's the one wearing the England football top for those that are listening. that's I just love the humour, and there's a place for humour. And back at Essex, we used to have an annual play similar by the postgraduate students that ripped all the academics off. Any faux pas they'd made during the year would come back and haunt them each year.
Yes.
It was good for team spirit. It was good for morale. It was good for breaking down barriers.
Yes.
Throughout it.
And as time went on, my skits became less about lampooning the senior figures and more about the general plight of people in research. And I had a version of the Beatles' Penny Lane, which was about the research student throwing their chance and having their last turn of the roulette because of the tremendous risk of doing research. And I actually had some people in the audience actually in tears over that. So the skits had sort of morphed into something more communal and more less of a comedy show. But there we are. But I did do other things. It was only 1 month in each year that I devoted to.
You talked enough about evenings in the lab making lenses and samples and test slides. So we do. Don't worry, Brad. We're not thinking you're spending all your time doing skits. You sent me this picture, and this is a complete change of subject. This is your drawing, I presume, of a.
Correct.
Oh, you tell us what's it a drawing of? Because I'll get it wrong.
This is a squirrelfish, an endemic species of Ascension Island, which is one of the string of islands that runs across the Atlantic.
And you did this yourself?
I painted it for Roger Lubbock, who was a zoologist, because after my zoological phase, when the Danish professor died, I became more zoological because of striking up an acquaintance with an ichthyologist who was extremely brilliant and had been misguided by his school. The school had sent him to Oxford as a historian, not realizing that he was a scientist, even though he had kept tanks at school and was obsessed with fish and went scuba diving and so on. You may have heard of the school. It's called Eton College. But they got this completely wrong. And Christchurch in Oxford said to him, "Do you realize that you're a scientist? Go away, do some A-levels in scientific subjects, and then come back and we'll look at you." So they saw something in him, and it was actually considerable intellectual brilliance. When he arrived in Cambridge, I was in my state of despond because of the death of my protector, in a way. And Roger turned up, and he started asking me things. He was a confident Etonian. So if he wanted to know something, he just went and asked somebody, had no compunction about disturbing them or anything. Very, very confident. And I ended up being his unofficial supervisor and teaching him about science and about, if you can believe it, mathematics even. And I remember he came to me once and he said, Brad, these exponential logarithms that you're telling me about, they're bloody good, aren't they? Well, why did they teach me about logs to the base 10? Because the exponential ones are so much better. Which makes him sound like an idiot, but he wasn't no idiot. He got a personal MRC grant to study self, non-self distinction by coelenterates. And he taught Linda, my wife, my late wife, and me how to scuba dive. And we ended up.
These notes.
Yes. Yeah, this is a picture of I'm the one standing up in the outrigger canoe, and this is off the island of Palawan in the Philippines, which is one of these places that you must go there if you're a zoologist or a shell collector or anything like that. Palawan in the Sulu Sea is a place to conjure with, and
wife, I think,
if you go back to that, can you go back to the previous?
Yeah, of course I can.
Slide. The person in the sea in a scuba outfit who you can't see, but who took that picture was my wife. So she and Roger dived in the Philippines, and I couldn't because I developed a tropical head cold, so I couldn't put my head more than 1 meter below the surface of the sea without intense pain. So anyway, the next picture shows Linda on a holiday that we had in the Maldives. She's on a crowded dive boat, and that's her in the foreground with the yellow adjustable buoyancy life jacket. And she was incredibly brave because she already was showing some return of the multiple sclerosis, which appeared in the earliest years of our marriage. It was coming back, but she still learned to scuba dive, and she dived with me. We dived off that boat among sharks because the guy standing up, Helmut Voigtmann, was a crazy German who liked to go down and holding fish in steel reinforced gloves would feed them to sharks. So we sat, Linda and I, on a sandy bank at a depth of about 5 meters, and he was waving these fish about, and these reef sharks came around, and you didn't see them bite. You simply saw them cruise by. There was a flash because they jerked their head up faster than you can really follow it by eye, and there was the fish halved and a little blood floating in the water. So this was completely mad, the whole business, but we did it. He just informed us beforehand that if the shark comes to you, you do not put out your hand. He just wishes to know if you are British or Spanish. If you put out your hand, he will take it. So we sat with our hands tucked in as well as we possibly could, and we survived.
You're braver than I would be.
But I did nearly kill myself once scuba diving, but that's another story.
Back to the fish, because
Yes.
this fish, I believe, from the pictures you sent me, has another story as well behind it. Or is
Yes,
that another fish?
that's right. Well, I was working in the lab, and two extremely polite gentlemen in tweed suits turned up. And they explained that they were both company directors who had retired and were now working as Crown agents. So they were working for the Queen, and they were responsible for the design of British postage stamps. And they wanted my painting. The painting that's behind you is one that they wanted to use on a stamp for Ascension Island, and it's the bottom left one. That's the squirrel fish. So I got that first day cover and the picture returned, and that was my my royal duty executed. Unfortunately, although I thought at one time I might be a painter, my painting has been and drawing has been really reduced to illustrating Roger's ichthyological things and diagrams for scientific purposes. And at the start of the confocal project, I was using all of my faculties. When Bio-Rad started to manufacture, because Bio-Rad were not terribly interested in putting any money into it. So John White and I had to really work hard to make sure that the thing succeeded. Together, we worked on the production line to get systems shipped off to the USA. And I was drawing from then on quite a lot, because I had to do the engineering drawings for all of the scan head components, all of the adapters to fit the MRC 500 and all the other microscopes to different microscopes. So I would take my drawings, which were, there was no CAD in those days, or it was barely available. So there were Indian ink on faint blue graph paper, and I would go to all of the subcontractors, and all too often I'd be discussing the drawing with the subcontractor, the guy who was actually going to carve metal, and the financial person in the company would come in and say, "We love to talk to you, Brad, but I'm afraid we can't do it anymore because Bio-Rad have not paid their bills." So the company was really nickel and diming all the time.
Yeah.
There are many stories about the president of this international company who was so pathologically mean that he influenced the whole outfit, and there was nobody hired for R&D for 2 years. So I was the R&D, I was the promotion, making 2 Far East trips, which could be quite long, 3 USA trips and several Europe trips in a year. And I got the opportunity to learn about American and Japanese, Korean and Thai culture. So it was very interesting, but also tremendously exhausting, because I was also having to write patents at the time. And my wife said, you're able to live like a Renaissance man because the company is such crap. And that really was the situation. Later on, they slowly began to hire people, but the basic problem was that they were interested in consumables and not in
The equipment.
selling instruments. The secret of the company's success was making HPLC kit, but that trapped every hospital into buying the consumables, including standard urine and other things perpetually. So they weren't so keen on British people. They weren't so keen on microscopes either.
So thinking about traveling to all these places, some quick fire questions. What's your favorite food? Been to all these places, what is your favorite food? If you were to be taken out for dinner, what would you order?
I was terrifically fond of sashimi and the tuna and the shrimp and so on that I had in Japan was always fantastic. I confess that I also tried whale meat. Now, my family, when they had lived in Liverpool during the war, had been obliged to sometimes eat whale meat. It's totally disgusting, auto-digested rubbish. But the whale meat that figured in Japanese cuisine was absolutely delicious. And of course, I used the rather feeble moral excuse that the whale had already been killed, so it made no difference whether I ate it or not. But anyway, it was delicious.
That's never going to hold out as an argument, and we know that one. What's your least favorite food? Is there any food you really don't like?
There is one particular Indian dish which is called murgi mosolia, and it makes me throw up. So I think it contains some antigen to which I'm allergic. I have no other food allergies at all. It's just that one particular dish.
Okay. Are you an early bird or night owl?
I tend to slide later and later. Yeah, that causes problems because you have to just stop it eventually and try and reset yourself.
Okay. That's interesting. Tea or coffee?
Oh, coffee. I have a vast consumption of coffee, and yes, I used to grind it, but now I use pre-ground coffee, so I lowered my standards a little bit. But for
there's a place for everything in this world. There's a place for everything still.
Actually, you can improve the taste of coffee by adding a tiny touch of salt, and there's a Viennese trick where you add a tiny little bit of chocolate to the coffee before you allow it to percolate.
Okay, I was going to say, what type of coffee then at that point? Beer or wine?
Sorry?
Beer or wine?
I confess I've kind of taken to having a Stella Artois in the evening, and I used not to be a beer drinker at all, but I find that it's very convenient, and I just go to the fridge and I get my Stella. Wine drinking is difficult if you're on your own. I'm now living with my younger son, who does not drink wine, and so it's quite difficult. I love especially Burgundy, and I like the Rhone wines as well. But I don't really get much chance to drink. It would be a solitary occupation, which would be no fun.
Okay. Chocolate or cheese?
Chocolate or
cheese?
Cheese.
So sweet or savory, I guess.
Yes. Yeah. I'm very partial to a nice brie or a Stilton. I think I do get easily sickened by chocolate, because I tend to eat the lot in one go. But cheese is one of my gourmet treats.
You should go dark chocolate then. It's always by my side. You don't tend to gorge so much, just snack on it. Mac or PC?
PC, because as soon as you start working with a company, you tend to have to use a PC. The software for the original confocal from Bio-Rad was an IBM 286 PC, because Richard Durbin, who's now he was head of the World Human Genome Project, but he wrote the software as a favor to his supervisor, John White, and he tried to use the Mac because everyone in the LMB uses a Mac, but he found that Apple were too secretive about their operating system, and he had to follow the pattern of industry in using the PC.
Okay. McDonald's or Burger King?
Try to avoid both, I think.
Okay, that's a fair enough answer. Book or TV?
Sorry, which or TV?
Yeah, book or TV?
Oh, book or TV. I don't watch TV very much now, except for the news. And my taste in books is something like Joe Nesbø, Scandi-Noir, when I'm just wanting to do something and have a horror story from Norway instead of thinking. But I've tended more to read non-fiction, and I'm very impressed with the William Sargent book, which I've looked at again and again, actually all my life, called the Battle for the Mind, because I think it's highly relevant to our current situation in the world and in politics. Sargent was I don't know if you know the book.
No, I don't.
He was a Cambridge psychologist who dealt with World War II shell shock victims and people who had suffered brainwashing and terrible tortures and so on. And he put together this book which examines how the human mind can be washed, and the prerequisites for successfully getting ideas to penetrate into the human brain. And this has become a major interest for me. It's maybe not appropriate, but to summarize briefly, Sargent found that all religious cults, including things the Laplanders, North American Indians, Catholic liturgy, and other things, had the same quality of rhythmic chanting at about the same frequency, about the frequency of the human heart. And he explains how when you have fear in your mind is very susceptible to suggestion, and you will accept things which are manifestly untrue. They would not be accepted by the thinking side of your brain. And I have a feeling that we need to do that kind of analysis. There are interesting scientific experiments in experimental psychology that I could describe, but I won't. But the situation now is that every country in the world has more or less simultaneously given itself over to strong men. At the same time, we have the notion of climate change, and that the synchrony of all of these events all over the world in different cultures suggests to me that human beings are afraid now, and that they have become susceptible to political persuasion. And I think there is a part of the brain which is particularly non-logical and is a receptor. And to have it, I think, is actually adaptive, because we now know that our closest relative, the chimpanzee, is not like other monkeys. It's very like us, however, because the males go out in a formation, a military formation, and they go around the perimeter of their territory and they murder any chimpanzee, and occasionally other monkeys as well, who comes within their territory. This is exactly what we do. And this is why humane, civilized, cultured nations, which have parliaments, even with women MPs and so on, can become fascist, as happened in Weimar Germany. So you can have the logical brain, but you can also have within it this capacity to commit genocide. And I think that it's actually, as a zoologist, you've got to explain everything in terms of evolution. And I fear that we have this built into our brains so that we can receive things like war cries and act on them. And the word slogan is interesting, because political slogans, I think, can go into the brain with this William Sargent type conditioning. The word slogan is actually Scottish Gaelic and Irish Gaelic for a war cry. And
I didn't know that.
I've gone on too long about this issue, but
we can't end on that somber note. So, and we've gone over the hour by a few minutes, actually. So I'm going to ask you a couple of other quick questions. Are you a tidy or a messy person?
I am grotesquely messy, with terrible long periods when I vow to be tidy. I tidy everything, and it takes me an hour to mess it all up again.
And do you have any bad habits?
The old man's tendency to blether, which is roundly pointed out when I go to Scotland, but that's good for me to have it, to be reminded.
So, Brad, there was a couple of things that we didn't get through, but it is up to the hour. I've got to get, because I love this picture. This picture you sent was when you were, I think from the text you sent me, was when you were 14. Is that correct?
Yes, and it represents one of my mistakes, because I thought that this was science. I thought that a beautiful drawing was actually doing science. I didn't realize that for research, you very seldom need beautiful drawings. So I got it all wrong. I neglected my maths. I just didn't understand.
And it was, but it was a microscope picture when you were 14. It was through a microscope.
Yes, it was taken with a brass size microscope with a British-made low-power objective stuck on it. Yes.
It's a stunning picture, and at the age of 14, which is amazing. Brad, thank you so much for taking your time today, because I very much appreciate it. I look forward to being part of your next skit, which no doubt might end up being the case. Thank you, everyone, for listening, watching this episode of The Microscopist. Please don't forget to subscribe to whichever channel you're on. And I hope we get Gail soon on this.
Yes.
Brad, thank you very much again.
Thank you for having me.
It's more than a pleasure. Thank you for agreeing.
Thank you for listening to The Microscopists, a bite-sized bio podcast sponsored by Zeiss Microscopy. To view all audio and video recordings from this series, please visit bitesizebio.com/the-microscopists.